Hi guys. So you wouldn't believe it. I literally just did an entire lecture and didn't record a thing. So lucky us, I get to do it again. So macromolecules. What are they? What do they do? And how do they form? So this will be a little bit of a review for some of the items here, especially with what is a polymer. So we went over monomers and polymers. Remember my brick versus my brick wall example? Monomers, mono means one, poly, many. So a monomer bonds together to form polymers. Polymers break down into their individual monomers. We need monomers to build polymers. No getting around that. And how do these polymers form? It's through dehydration synthesis. So dehydration synthesis, we've gone over. You've already quizzed on it. Most of us have got it locked and loaded. Dehydration synthesis is the removal of water. So we can see here, I've got H and OH. I have two H's and one oxygen. Two hydrogens, one oxygen, H2O. That's how we remove the water. So when we do go through dehydration synthesis, a water is removed, so it ends up in our product section, and we form a polymer. You could see here, I go from two glucose molecules to one maltose molecule. How are they broken down? Well, they have to be broken down because remember, your cells do not thank you for the cheeseburger that you ate. They thank you for all of the monomers, all of those amino acids, those fatty acids, those monosaccharides that you get from your food. So when we add water to a polymer, we break it back down into its original monomer. So you can see the reverse happening here. Carbohydrates. You guys did a quick CK12 on carbohydrates. Most all of us turned it in. This is your reminder to turn it in if you have not yet. So carbohydrates are made up of carbon, hydrogen, and oxygen. However, they go in a very specific ratio. For every one carbon, there's two hydrogens and one oxygen, which is why my formula here is CxH2xOx. For example, glucose is C6H12, two times the amount of carbon, O6. So it follows our formula. Carbohydrates are used for energy. They're used for structures in plants like cellulose. And they're also used for blood glucose and glycogen, which is in our muscles, gets broken down for energy during hard workouts or long enduring workouts. The monomer for our carbohydrates is called a monosaccharide. Mono meaning one, saccharide meaning sugar. Our typical monosaccharide for carbohydrates is glucose. When glucose goes through dehydration synthesis, it forms bonds with other glucose molecules able to form a starch, glycogen, cellulose, or chitin, which is another carbohydrate structure. So carbs, important for energy, important to be broken down, and they help plants with their structures. Because remember, plants don't have skeletons. It would be weird if they did. DNA and RNA. You guys probably talked about this in middle school. You probably talked about your A's, your T's, your C's, your G's, or you at least talked about DNA and genes and where is it. So DNA and RNA are part of a group called nucleic acids. Nucleic acid is the polymer. Our monomer is down here. It's a nucleotide. A single nucleotide has a phosphate, a sugar, and a nitrogen base. Now, it depends on what that sugar is and that nitrogen base, whether or not it's DNA and RNA. DNA and RNA have three major differences between them. DNA is double-stranded, as we can see here. It uses deoxyribose as its sugar, and it uses thymine, cytosine, guanine, and adenine as its bases. Now, these bases form hydrogen bonds between each other, adenine with thymine, cytosine with guanine. And they always stay in that order, A's and T's, C's and G's. Now with that, you have RNA. RNA is like a messenger. So what it does is it copies the message from DNA and it's allowed to leave the nucleus, unlike DNA. So RNA is single-stranded. It uses ribose as its sugar. And instead of thymine, it will use uracil. So DNA and RNA share lots of things in common, but they have those three main differences between them. And right now for us, it's important to know that they are made up of nucleotides. They build to nucleic acids And there's 2 different nucleic acids that we are talking about. And last but not least, our false polymer. So lipids are not true polymers. They do not have single monomers. A small lipid is something like a glycerol and 3 fatty acids, which makes up a triglyceride molecule. These things are made up of carbon, hydrogen, and oxygen, which so are carbohydrates. However, these ones can come in 2 fashions, and they don't have a specific ratio. So when we look at lipids, like foods that we consume, we kind of can separate them into saturated and unsaturated fats. Saturated fats have single bonds between the carbon atoms, which means each carbon has the maximum amount of hydrogens that it can around its atom. And remember, carbon can hold hands with 4 different atoms. So every single carbon in a saturated fat has 3 hydrogen atoms attached to it. And the way that we can tell saturateds versus unsaturated is real easy. Saturateds are solid at room temperature. Our unsaturated fats down here, or over here, I should say, have a double bond. So not every single carbon has 3 hydrogen atoms. They may only have 2. And having that double bond causes a little bit of a wiggle in the structure, and it won't solidify at room temperature. In fact, they stay liquid. Things like olive oil, vegetable oil, corn oil, grapeseed oil, avocado oil, all of those are unsaturated fats because not every single carbon has 3 hydrogens attached to it. And last but not least are phospholipids. These make up our cell membranes. These are super, super important. So our phospholipids have a hydrophilic head, which means they bond to water, and they have hydrophobic tails, which means they don't like water. So your cells have to have this membrane. This is what keeps things in and helps keep things out. Think about it as the bouncer of the cell, right? So our hydrophilic heads and our hydrophobic tails will arrange themselves so that the heads face the insides and the outsides of your cell, and they make like a little phospholipid sandwich. The tails stay nice and dry, whereas the heads form bonds with the liquids inside and outside the cell, because remember what I said, you guys are wet on the inside. So I do hope this was short. I do hope it was sweet. Please pause, go back,